presenting to you the cell cycle. Interphase is the beginning of mitosis, when the cell grows and performs at its normal function. Interphase is subdivided into G1, S, and G2 phase. To start out, the blue piece along here is the cell, and this is the nucleus here, the pink thread. G1 is known as the first gap or growth phase in the cell cycle, which the cell is not dividing. Majority of the cells are in interphase most of the time. The S phase, which is synthesis phase, is when a cell replicates its DNA, and that lasts about 8 to 10 hours. The amount of DNA within the cell, it doubles. G2 is when the cell continues to grow and make preparation for mitosis.
Prophase is the first phase of cell division, and there are forming of the X-shaped sister chromatin nucleus. The nucleus disappears, which is this, and they're on one end of the cell, and the others are on the other end of the cell.
And next, we are going to start the metaphase. The metaphase is where the chromosomes align against the metaphase plate, which is equal distance between the two spindle fibers. And they align and they attach to the kinetochore right here and the centromere right here. And they are pulled together by the spindle fibers from both sides. That's it.
In anaphase, the paired chromosomes, also known as the sister chromatids, separate and begin moving to opposite ends of the cell. Spindle fibers not connected to chromatids lengthen and elongate the cell. At the end of anaphase, each pole contains a complete compilation of chromosomes. The paired centromeres in each distinct chromosome move apart. Once the paired two sister chromatids separate from one another, each is considered a full chromosome. They are referred to as daughter chromosomes. Through the spindle apparatus, the daughter chromosomes move to the poles at opposite ends of the cell. The daughter chromosomes migrate centromere first, and the connected core fibers become shorter as the chromosomes near a pole. In preparation for telophase, the two cell poles also move further apart during the course of anaphase. At the end of anaphase, each pole contains a complete compilation of chromosomes.
Okay. Then the last phase, which is the fourth phase of mitosis, is telophase. It's defined as a stage of mitosis when the new daughter chromosome cells change back into the new threads of chromatin, and the new nuclear membranes begin to form. Also during telophase, new nucleoli appear in newly formed nucleuses as the final stage, and concludes cytoplasm dividing in half as the cell membranes begin to close up around the new daughter cells. This final process of cell reproduction, which is cytokinesis, is defined as division of cytoplasm during mitosis. That takes about 10 to 15 minutes. It's required for telophase to finish, and also cytokinesis. Whereas the entire cell reproduction process is completed, and it returns to the interphase state. So we should have bundles, spindles on the end, and each possesses two identical genes of its parents. And there are two new half-size cells that will grow larger until each achieves the size of the parent cell, of the original parent cell, and that's telophase. So we have two daughter cells right here.